Monoclonal antibodies in myeloma, as we discussed earlier, have been really a breakthrough in the treatment of the disease. How they work is actually fascinating. They target very specific antigens expressed by the tumor cell, classically on the cell surface. In this case, our major classes target either CD38 or this other entity called SLAMF7, which is also used to be called CS1 previously. These antibodies do a number of things. And if we focus on the CD38 space, daratumumab is obviously the most important example. In terms of how daratumumab works, it does so in a variety of ways. It activates the immune system on the one hand, and at the same time, although this is a very important mechanism to appreciate, it also triggers what we call programmed cell death in the tumor directly. So it has what we call an apoptotic mechanism, as well as a variety of ways of activating the immune system. One of the interesting ways it activates the immune system is through complement activation. Another actually is through a variety of cell-mediated mechanisms that can attack the tumor. So because of that, that's why, for example, you have to have pre-medications when you have daratumumab, because it activates the immune system, so you mustn't have that be too robust. But at the same time, what we've also learned is that it combines very effectively with other drugs, and that there's true synergy with these other drugs in terms of the way they work. Now, another monoclonal antibody we touched on before, elotuzumab, which targets so-called SLAMF7, is more of a pure immune stimulator. It simply activates natural killer cells, but at the same time targets myeloma, brings them together, and when you put into that space an immunomodulatory drug, like either lenalidomide or pomalidomide, this whole partnership is highly synergistic. So that's broadly how antibodies work, as two obvious examples and two approved examples in this setting.